University of Stony Brook. In 1981, he trained in the National Cancer Institute Medical Oncology Fellowship Program as a board-certified internal medicine and medical oncology physician. Dr. Frank became a tenured senior investigator at the National Institute of Health in 1989 and is currently the chief of the Laboratory of Diagnostic Radiology Research and the Frank Laboratory in Radiology and Imaging Sciences at the NIH. The Frank Lab is primarily focused on the development of techniques to improve cell therapy for regenerative medicine by using cellular and molecular imaging to track cells, and also, as the topic of today's presentation, use of pulse-focused ultrasound to enhance homing, permeability, and retention of cells in targeted tissue.
Thank you. Thank you for the invitation to speak today. I'm going to be talking about how we have been using focused ultrasound, pulse-focused ultrasound in enabling and enhancing the effects of cell therapy. When we look at stem cell homing and how cells get to your tissue of interest for regenerative medicine or even for immunotherapy, they tend to migrate through the vasculature and then they either stick by tethering and rolling onto cell adhesion molecules on the endothelial surface in the area of inflammation and then transmigrate through, sort of active homing, or there's passive homing where they actually stick into the vasculature and then try to transmigrate through into the parenchyma. And there are certain ways to enhance homing, which is you can directly inject your cells into the tissues. You can magnetically direct cells into an area. We did that years ago where we showed that if you actually label a cell with iron oxide nanoparticles and place a magnet externally to where the cells are, you can actually get them to stick into an area and increase the numbers. You can modify the cell surface or modify the cells so that they produce integrins on their surface to actually stick to the areas of inflammation. And therapeutic ultrasound, we do know, increases angiogenesis through the heat and basically stimulating VEGF secretion. And we're going to add to this now pulse-focused ultrasound. What's interesting is with stem cells, so this paradigm tends to be used for leukocytes, monocytes, T cells, dendritic cells, but it can also be applied to stem cells. And the reason why it can be applied to stem cells is because we actually have integrins on the surface of the cells, much like on T cells and monocytes and macrophages, that actually can stick to the cell adhesion molecules, the CAMs or the CAMs, on the surface of the activated endothelial cells. So this is the common paradigm we see with pulse-focused ultrasound. You turn on and off the ultrasound beam very rapidly, in this case 10% duty cycle, and you get a 4 to 5% increase in temperature. That's different than continuous focused ultrasound, where you get ablation, very high temperatures in a very short period of time. In our case, we tend to use 5% or less duty cycles. And how did I get into this field is really in a different sort of way than my background would be in MR and actually in cell therapy, is that I started looking at some of the results coming out from Viktor Frankl and King Lee's lab and realized, in fact, that in the pulse-focused ultrasound treated muscle, you got increased albumin leakage into the extracellular space. This is really important because years ago in my career, I studied muscle physiology and muscle exercise physiology just briefly and realized that, in fact, that increased extracellular space that you're seeing around the muscle is similar to what happens when you strenuously exercise. So if you go out for a long run, you do a lot of weights in the treatment room, you actually get this effect. And so we actually went back and looked at this and asked the question. People are using pulse-focused ultrasound to enhance drug delivery, enhance permeability and retention of drugs and genes to a tissue. And in this case, tumor tissue, you see the increase in extracellular spaces after you treat the tissue with PFAS. If you actually inject, and Victor did this work, inject the dye into the vasculature that stays in the vasculature, after PFAS, it leaks out. It leaks out, the cells become leaky. So if you can use, and this is the basis for this whole project, if you can use PFAS to increase the EPR effect in tissues for gene and drug delivery, could you actually use the same effect to increase stem cell homing? Very simple process. Can we go from drugs moving into cells? And so our first experiment, we actually gave a fluorescent iron oxide nanoparticle to the animals before, 3 days before. We know we could label the splenocytes, the macrophages in the spleen, and we could tell that they're actually there, and they're Prussian blue positive, and they are fluorescently labeled, and we didn't see any liver. And then we just did this simple experiment, compared continuous HIFU to pulse HIFU, knowing that, in fact, we wouldn't cause damage with the pulse HIFU settings in the 5% duty site. And what we found on MR is that the cells, and day 3 and day 8, there are a lot of black dots here, which are basically macrophages filled with iron going to the damaged tissue, but we could also see the same effect with PFAS. When we went and looked at histology, which was really the most important part of this experiment, we could find our Prussian blue positive macrophages in the tissue with PFAS, as well as with continuous focused ultrasound or ablation. You could see the ablated muscle here, but we could actually know that those cells came from the spleen because they were fluorescently labeled on histology. The fluorescent macrophages were here in orange, but we also got increased signal intensity of the cell adhesion molecules on the endothelial cells in the muscle. One part of the equation that I showed you in the first cartoon was the fact that you needed increased cell adhesion molecules on endothelial cells to get cells to stick to an area. We then went ahead and looked at the cytokine profile. And this hadn't really been done. This is 1 day later, and we found that there were both pro-inflammatory and trophic factors being elevated in the focused ultrasound treated tissue, as well as in the ablated tissue, compared to contralateral control. And some very important factors such as SDF1 stromal-derived factor 1, which causes stem cell homing. So now we have 2 points of it. We have a gradient here that we see. We have cell adhesion molecules, and we ask the question, could we actually do this study? Was pulse focused ultrasound causing a mechanotransductive effect, a mechanical effect that's causing a biological downstream effect in tissues releasing certain cytokines, chemokines, and trophic factors, CTTFs, and cell adhesion, and upregulating cell adhesion molecules in the muscle that could increase cell homing? Simple question, can we do that? And so this is the model. Can pulse-focused ultrasound actually increase stem cell homing? And so when you normally inject stem cells into tissue, they travel to the vasculature and they don't see any damage. They just continue to travel and they ultimately disappear. Within a relatively short period of time, by a week, most of the cells, more than 97% of the cells, cannot be found in the tissue. If you now pulse an area, you now got an increased regulation of upregulation of chemoattractants, cytokines, chemokines, and trophic factors, cell adhesion molecules, and what we have called in terms of transient molecular zip code in the area. This is a place where the cells want to get off the bus. So now you go ahead and you inject your cells, and you find out that there are more cells concentrated in the area. And we call this, instead of an EPR effect, we call it an enhanced homing permeability and retention effect. We've made the environment such by pulse-focused ultrasound so that the cells want to actually get off and transmigrate into the tissue and stay in the tissue. Capillaries, venules, arterioles. So we went back and did a simple experiment design. We wanted to do something deep, not muscle, which is fairly superficial. So we went to the mouse kidney, partly because we had an in vivo built-in control. And we could localize the kidney very nicely. We could pulse in the kidney in certain areas. And we could then harvest the animal and harvest the tissue at various different times, do a variety of molecular techniques, as well as imaging and look at histology. And the first thing we did is we labeled our mesenchymal stromal cells, or mesenchymal stem cells from humans, and we were putting it into mouse. And we actually labeled them with iron oxide, and we injected them to the animals after PFAS about 4 hours after PFAS, and we looked at the kidney, and we could find a lot of Prussian blue positive cells in the kidney, in the peritubular spaces, mostly not stuck in the glomeruli, but peritubular spaces. And this is a contralateral control, and we could see an effect on MRI. We went back and looked at it with a human mitochondrial, so we're actually looking for human cells in a mouse model. We could see large numbers of these purple cells outside the tubules in the kidney. This is a magnified view. We could also see cell adhesion molecules upregulated on the kidney endothelial cells, the vasculature. So we get cell adhesion molecules coming back up, as well as the endothelial cells, and we could find some interaction of our red cells with the endothelial, the cell adhesion molecules on the surface. We went ahead and did a count, and on day one day after we gave the cells and treated, we found about an 8-fold difference in the number of cells in the tissue. 3 days later, we found about a 5-fold difference in the number of cells in the tissue, in the pulse-focused treated tissue compared to contralateral control, and they were all gone by 7 days, which is pretty consistent with what the literature would say, but initially we found a large amplification of the number of cells within the tissue. And also cell adhesion molecules were. We went back and looked at the cytokine profile, the molecular changes in the tissue, and once again we had both a combination of pro-inflammatory and anti-inflammatory and trophic factors being upregulated in the post-focused ultrasound treated kidney, even out to 1 day later, compared to the contralateral control. And that if we gave the cells and then harvested the tissue 6 hours after we gave the mesenchymal stem cells, we actually downregulated the effects of the pulse-focused ultrasound. What does that mean? Well, mesenchymal stem cells are an immunomodulatory, decreases inflammation in tissue, and therefore we're decreasing the effects in the tissue. So we went and decided that given that this was all being done in normal tissue, we wanted to look what happened in the disease model. With my oncology background, we wanted to look at acute tubular necrosis. This is acute kidney injury in a mouse. And we wanted to treat only one kidney that in the mouse with pulse-focused ultrasound and give stem cells to the animal. We treated the animals, pre-treated the animals with cisplatin. 1 day later, we treated with PFAS and cells to one kidney, or on 4 days later, we gave PFAS and cells. And we killed the animals 1 day later. And what we found, again, is increased numbers of cells in the PFAS-treated kidney. This is with acute kidney injury compared to the contralateral control. We do have some passive homing by day 5 of the cells actually leaking out of the tissue, but this is still statistically significantly different. And we can see that. What's most remarkable is we didn't really understand whether or not PFAS was actually going to make the disease worse or actually keep the disease or make the disease better alone. So could, in fact, focused ultrasound make the disease worse or make it better? Because all we had done is actually study normal tissue. And so we actually designed this protocol where we treated both these now in an animal that has received cisplatin, so we had acute tubular necrosis going on, and we gave them cells. And first we needed to look at the natural history of what's going on in the tissue, and this is the molecular changes. And first, essentially by day 1, most of the cytokines are down in the tissue. Most of the trophic factors are down because there's a lot of destruction going on in the tissue, and slowly they come back up by 7 days. When we pulsed an already diseased kidney with active acute tubular necrosis, acute kidney injury on either day 1 or day 4, what we find is some of the factors actually also get upregulated. So whatever cells are still healthy or just barely holding on actually still will secrete some effects as a result of pulse-focused ultrasound. What's particularly interesting is we can get an upregulation of interferon gamma. And I'll come back to this later, because interferon gamma with TNF-alpha and IL-6 actually can each potentiate the effects of MSCs, can potentiate the effects of mesenchymal stem cells in the tissue. We know, and I'll just go quickly through this, that we actually saw no effect of pulse-focused ultrasound as far as causing damage in the acute kidney injury model. So it doesn't potentiate the effects of the injury. It doesn't make them worse. But what happens is when we gave the cells in acute kidney injury on day 1, we wound up with pulse-focused ultrasound. we wound up getting improvement in a variety of different things. So first of all, we saw improvements in renal function. So the BUN and creatinine in the pulse-focused ultrasound treated kidney with stem cells wound up more like normal as compared to the cells alone. So the cells, there were statistically significant differences between the cells versus the PFAS-treated kidneys with cells for both serum creatinine and BUN. We also found out a variety of different markers, survival markers, phospho-AKT, proliferation markers, Ki-67, decreased apoptosis in the kidneys that were treated with PFAS and MSCs compared to MSCs alone, decreased necrotic tubules. We also then went ahead and looked at what happened to the immunological profile in the tissue, and so we looked at macrophages. So there's a concept in the literature of an M1 and an M2 macrophage, M1 pro-inflammatory macrophage, M2 anti-inflammatory macrophage, and what we found is PFAS with cells increase, as well as cells increase the number of anti-inflammatory macrophages within the tissue. Decrease NK cells, decrease dendritic cells, increase PFAS plus the cells, increase the vascularization of the kidney compared to the MSCs alone. So again, we're seeing a variety of factors that are suggesting that we're trying to heal this kidney that's now in acute damage, and that we could potentially prevent the disease. I think this was the most remarkable thing that came out in our paper in Stem Cells in 2015 by Scott Burks, is that we decided to delay the treatment effects of MSCs and pulse-focused ultrasound to 3 days when there's full-blown acute kidney injury going on in the animal. And in this case, this experiment hadn't been done in the literature. Most cases, the experiments are done on day 1 or day 0, where they give cells to prevent kidney injury. In this case, it was really a matter of survival. And what you see here in the white bar is just saline treatment of the animal. What you see in the yellow bar is with stem cells, and there's a statistically significant difference between the cells and the saline-treated animals. We also see a highly statistically significant difference between PFAS and cells compared to cells alone. So we actually improved the survival. We were able to rescue a dying animal by giving the combination of cells and PFAS versus cells alone. This had not been reported before. We got asked by the reviewers what was the mechanism was going on, and actually was a very important question. So we redid the experiment again. And remember, I told you that interferon gamma went up in the acute kidney injury model following PFAS. What we found is that in this model, where we killed the animal 1 day after doing PFAS plus MSCs, we found that because we're giving human cells, IL-10 went up. This is an anti-inflammatory cytokine. We found a decrease in tumor necrosis factor alpha, mouse tumor necrosis factor alpha, and in the kidneys of the mice. And we saw increased VEGF and in the mouse serum and as well as in the kidney, suggesting in fact that we were getting some preconditioning of the tissue such that with the increase in interferon gamma, MSCs became more potent in that tissue. So that combination was more potent than just the MSCs alone without PFAS. And really one of the questions that come up is, can PFAS be used to precondition and serve as a neoadjuvant before you give your cellular therapy? It could also serve as a neoadjuvant approach to basically giving immunotherapy. Can you pre-treat this tissue with ultrasound and therefore get an enhancement, a local enhancement where you treat it with pulse-focused ultrasound? And our goal ultimately is to start looking at other models, including renal transplantation, because I think it's very important to see if we can downregulate the number of drugs that are being given in order to maintain the graft in the host, and possibly just use stem cells to treat a kidney. I'm going to talk about a few other studies here. I'm going to look at the effects of pulse-focused ultrasound temporally. What happens when you pulse and what happens afterwards? What happens with drug-host interactions and the effects on pulse-focused ultrasound. I'm going to show you 2 models of inflammation in the tissue. One's an MDX, or muscular dystrophy model in the muscle, and a chronic limb ischemia. So And I'm going to show you some very new data about what happens when you combine post-focus ultrasound and microbubbles. All of the studies have been done without microbubbles to date. And with microbubbles, you see the temporal changes. So we published this work in 2013, where we coupled post-focus ultrasound with either mesenchymal stromal cells or mesenchymal stem cells and endothelial precursor cells. These are cells that ultimately produce vessels in tissue. And we gave one pulse with the cells, and we then killed the animal 1 day later. Or we gave one pulse with cells and then gave 2 more courses of cells. Or we gave 3 combination cells plus T-plus on 3 different days and euthanized the animals later. And we wanted to just look at the number of cells in the tissue. Could we really enhance permeability and retention of cells in the tissue? And what we found in the first example, we found an increased number of cells in the tissue with both the endothelial precursor cells and the mesenchymal stromal cells. But more importantly, we got a massive amplification of the number of cells if we treated 3 days in a row compared to just the other 2 paratimes, 1 cell plus 1 level. So this is about 8 to 10-fold increase in the number of cells, and we could really see that, in fact, we are getting more cells when we do sort of this metronomic type of approach, this daily sort of PFAS coupled with stem cells being treated in the tissue. We also saw changes in cytokine profile, slightly more different type of effects. This is very early on, so we actually saw some anti-inflammatory as well as pro-inflammatory and trophic factor changes, as well as some very important chemoattractants that started to come up with 3 courses of PFAS compared to a single course of PFAS, including stromal-derived factor, stromal cell-derived factor 1 alpha. We did not see any changes in heat shock protein 7. We're not thermally injuring the tissue. We're not stressing out this tissue at all. And I asked a very important question several years ago: what happens after PFAS? We all tend to sample at 1 day, 10 hours later, so all over the place. What really was happening in the tissue? And this had not been done in the literature, and this is part of a PhD thesis that's just been published in Stem Cells in April this year, where we actually pulsed the muscle and the kidney of an animal, or the kidney of the animal, and harvested the muscle at different time points and did a molecular analysis. In the second experiment, which you'll see, is we gave cells either before or after PFAS to see whether or not there was going to be an effect, whether or not if you pre-treated the animals with cells and then gave PFAS or afterwards. We then did the simple experiment based on essentially this first experiment of looking at what happens if you have drugs on board in the animal, and then the effects of drugs on stem cell homing. So, very interesting observation came up. This is a heat map of fold changes in the tissue, in the homogenized tissue. And within the first 10 minutes, what we see is an increase of TNF alpha, and then it remains flat in the muscle. Followed by some other cytokines, pro-inflammatory cytokines, as well as VEGF, followed by a nadir, which is what we actually see, and then some other chemoattractants and anti-inflammatory cytokines coming up. This is a total of 5 seconds. We do the 5-second, 5% duty cycle, 1 hertz frequency repetition, and this is 5% of 5 seconds of post-focus ultrasound affecting tissue characteristics after 48 hours. Large number of things. This was not expected. We did not see it. So based upon this box diagram, where we see a lot of cell adhesion molecules coming up, a lot of anti-inflammatory changes going on in the muscle. We then use some cancer analogies to look at actually rheumatoid arthritis and see what happens when we plug TNF alpha as the inciting event here. What we found is that this is driving NF-kappa B and COX-2, cyclooxygenase 2. Cyclooxygenase 2 is something we tend to treat with ibuprofen when we go out and do strenuous exercise. So you tend to pre-treat yourself with anti-inflammatories because you don't want to have those muscle aches and pains for 3 days afterwards. Well, guess what? 2 or 3 days afterwards, or 2 days afterwards, you're getting muscle aches and pains because you're getting a lot of inflammatory cytokines in, as well as anti-inflammatory cytokines. So we went back and tested the hypothesis here. where we actually looked at COX-2 and NF-kappa B, and we found that within 30 minutes, but certainly peaking at 4 hours, COX-2, cyclooxygenase 2, was elevated, as well as NF-kappa B at 4 hours. And now we had a paradigm. Pulse focus ultrasound increased TNF-alpha, tumor necrosis factor alpha, transiently, drove essentially two other factors, nuclear factors and cytosol factors in the cells, and resulted in increase in chemoattractants and MSC homing. What happens if we use commonly available drugs to treat the animals and pre-treat the animals first, and then see what did we alter this paradigm? So we gave them ibuprofen, Motrin, at a normal dose that you would treat pain in an animal with, and Intercept, which is also used to treat rheumatoid arthritis in animals, in animal models, and we found that we suppress COX-2 and NF-kappa B being made in the cells. So we downregulated the biological effect, even though they all got the same post-focus ultrasound to the muscle. More importantly, you see in this heat map analysis, we by shutting down TNF-alpha, we actually wound up shutting down the whole cascade of effects from the drugs. More importantly, we stopped stem cell homing. So this is really important. White is the control animal, the control limb. These are all the control limbs. We also use the COX-2 knockout animal, a mouse that doesn't make COX-2, so that basically proves the point. And here's what happens when you treat an animal without drugs on board. This is what happens when you treat the animal with drugs on board. There's no statistically significant difference between the control and the post-focus ultrasound treated side, and there's a highly statistically significant difference, suggesting that, in fact, COX-2 and NF-kappa B were part of the pathways associated with and were being activated by post-focus ultrasound. We also did the experiment where we gave cells before or after, because there was some reference by Himmelman's group in the literature in PLOS ONE that suggested that there had to be ultrasound interactions with cells, which really, and we wanted to show that you didn't really need to have that interaction with cells. So if you have the cells on board before you do pulse-focus ultrasound, you get similar amounts of increased cell homing as if you gave the cells after pulse-focus ultrasound. And in fact, what you can give cells as much as 8 to 16 hours after you did PFAS, and you still get significant homing compared to the contralateral limb. So this is a 48-hour effect from 5 seconds of sonication radiation. Everything we have done so far has been in an acute inflammatory reaction. We wanted to see what would happen in chronic inflammation, because really the concept here is we can always give cells during acute inflammation. What happens when we come back after all the inflammation has died down and there's still a chronic disease, smoldering disease within the tissue? We looked at a muscular dystrophy model, which is a chronic inflammatory model in a mouse, and called the MDX mouse. Again, we looked at the profile following pulse-focus ultrasound with a 5% duty cycle. We're using about 4 megapascals worth of power. And we see, again, a lot of more inflammatory cytokines coming up over a 48-hour period. We actually found larger numbers of cells in the pulse-focus ultrasound treated tissue compared to the contralateral control. Even though there's ongoing inflammation, we were able to upregulate things in the tissue with pulse-focus ultrasound. More importantly, ibuprofen is used to treat patients with muscular dystrophy, and what we found is complete suppression of the stem cell homing to the tissue when ibuprofen was on board in these animal models. This is, again, what drugs you have on board may have an important aspect on what your cells, may affect cell therapy. Again, TNF-alpha, actually a lot of inflammatory cytokines came up very quickly within 10 minutes after giving PFAS. Again, a lot different profile in the kidney than we saw in the muscle. This is more of a Gaussian type of approach. Again, over now, even 72 hours, we can actually see an effect. We see a lot of inflammatory and anti-inflammatory cytokines, as well as cell adhesion molecules coming up. Again, we asked the question, if we block these pathways, any of these cytokines, the initial cytokines in the first set, COX-2, TNF-alpha, or interferon, or IL-1B, interleukin-1 alpha, we get a complete suppression of TNF-alpha, and we don't get and we have an impact on stem cell homing. So again, this is the untreated, this is the contralateral control. All the contralateral controls would then collapse down to this bar here, the white bar here. The drugs interfered with stem cell homing to the kidney. So basically, we have been able to show pretty much that we having a mechanotransductive effect, a mechanical effect, whether or not it's cavitation, acoustic cavitation, or radiation forces in muscle and kidney can drive a biological effect over a period of time, as much as 48 to 72 hours. That this effect can be interfered with drugs, so there's still mechanical stimulation going on in the tissue, but in fact, the biological effects that may be the downstream effects from the initial mechanical stimulation can be blocked with drugs, and can be blocked in chronic diseases as well as and in acute and normal tissue, and that we think we're getting close to a mechanism that calcium channels are involved. The stretch-activated calcium channels are involved, are what's causing this effect, translating the pulse focus ultrasound mechanical effect, mechanotransductive effect into the biological effect in tissue. We, as part of Pam Tabibi's thesis with a graduate student with Victor Frankel and myself, we started looking at chronic limb ischemia, partly because it's a muscle disease that we can actually get a handle on and it's easy to treat. It's associated with about 21 million people by 2020. It actually has a 2-year survival after amputation that's a very high. 2-year death rates, 20 to 40% of the patients will have a chance of amputation 6 months after a diagnosis. There are a variety of different ways that you can actually try to change the course of the disease, but most of them don't work because people don't stop smoking, they don't get the hypertension under control, they do take statins, and there's been a lot of things with gene and cell therapy by the treat the disease. And the other thing to realize is most of the literature in the animal models, they tend to treat with cell therapy very early, 1 day after the ligation, or day 0 when the ligation occurs. So we went ahead and looked at 2 different models and actually dropped the femoral artery. So this is lower down in the mouse compared to up in the groin area, which is the external iliac. We saw no change in flow because there's a lot of collateral vessels on laser Doppler after you ligate the femoral artery. But the external iliac artery, you actually get a decrease in flow. You see the limb missing below. What's remarkable is that if we left this process, so we ligate the external iliac artery and waited 2 weeks and then gave cells with PFAS and harvested the tissue, we actually found an increased number of mesenchymal stem cells in the targeted tissue compared to the contralateral control or just mesenchymal stem cells alone. What's most remarkable is that the combination of PFAS, 3 courses of PFAS with stem cells, compared to 3 courses of stem cells alone, the PFAS actually statistically increased the number of the blood flow in the tissue, in the muscle, and was compared to the contralateral limb, and compared to ischemic limbs that received only mesenchymal stem cells, PFAS alone, or saline. So here you can see a greater effect in flow as compared to 3 courses of stem cells, PFAS alone, actually this limb and this limb. What we also have seen is an increased number of vessels as a result of PFAS plus MSCs compared to MSCs alone. So we're increasing the vascularization in the tissue as a combination. We're changing the microenvironment because of the PFAS that's keeping the cells around to secrete factors that make more vessels. MSCs actually happen to secrete VEGF, which actually cause chemoattractant of endothelial cells and cause endothelial cell sprouting, as well as endothelial precursor cells coming out of the marrow. So we actually did see a tremendous effect in a chronic limb ischemia model. We saw a number of the vessels increase when we used PFAS plus MSCs compared to MSCs alone, highly statistically significant. We saw highly statistically significant starting 4 weeks after essentially the initial ligation. And the PFAS MSC treated group compared to the MSC treated group alone, we did not see that kind of effect when we did it on day 1. So if you treated the animals immediately after ligation, you don't see the effect. We don't understand why that is, but there's not a lot of other things that are going on that we don't really understand at this point and needs further evaluation. Summarize, how can we use PFAS for cellular therapy? Well, we're causing a molecular zip code in the area. We're causing a transient molecular zip code, which results in an increase in cytokines, chemokines, and trophic factors going up, as well as cell adhesion molecules upregulating on your endothelial cells. Coupled with cell infusions, we get increased numbers of cells, stem cells, as well as this would happen with immune cells as well. The same mechanism, these same factors cause immune cells to track to an area. And the big thing is PFAS without the cells on board does not alter the microenvironment, but does not change the cell product, does not change the cells you plan to infuse. Therefore, the path to using this combination through the FDA, even though you still need some advice from them, probably comes under the domain of an IRB modification to a protocol and not actually a full-blown IND. What other conditions can we do? Well, we're going to look at stroke, MI, chronic kidney disease, peripheral artery disease, and chronic limb ischemia, some more cancer, genetic disorders, transplantation. This may have a role in sports medicine, which we haven't really explored yet, but that all our muscle data would suggest that we may have a role there that we haven't seen before. It has a role probably in veterinary medicine as well, which we should explore. Tissue transplantation, organ transplantation is going to be very interesting to see. We want to see how we can use PFAS. We've only started to scratch the surface of sort of one course of PFAS and then cells. We don't know whether or not if you pre-treat the tissue multiple times with PFAS or focused ultrasound, and it then gave cells, would you actually enhance the effect? There's a lot of questions from a scientific point of view, how well it will translate. If you put more cells in the target tissue, will you get better results, or could you make things worse? There's a real question there. And I'm favoring that you could go back to this concept of doing a Monday, Wednesday, Friday type of treatment, especially if you can wheel the machine, the pulse-focused ultrasound machine, up to the bedside and do the treatment in the bed and just do the bag of stem cells and do it intravenously. And I think more importantly, we're starting to look at drug-host interaction. We think we're starting to get some idea about potentially how drugs are interacting with the tissue and what drugs not to have on board if you plan to use cell therapy with this. I think this is really important. This is my group at this point. So a lot of the work here was done by Scott Burks. He had some students, Michelle and Ben. Sophie Kovacs is doing the brain. Pam Tabibi is not in the picture, but she was she did her graduate thesis with us on doing the muscle and the temporal time course, as well as the chronic limb ischemia model. We get our cells from our cell processing section. We have a variety of different collaborations with the Kidney Disease Institute at NIH, as well as University of Minnesota with our muscular dystrophy model. And I'll take questions.